March 2020. Two patients lie in adjacent ICU beds, both struggling to breathe. Same age, same symptoms, same virus ravaging their lungs. One walks out 3 weeks later. The other never does. The doctors ran every test. Comorbidities? None that mattered. Treatment protocols? Identical. But buried in their DNA, invisible to every machine in that hospital, sat the real difference. A 50,000 base pair stretch of genetic code on chromosome 3. Ancient instructions written by a species that went extinct 40,000 years ago. The patient who died carried Neanderthal DNA, not as a curiosity or a footnote in some ancestry report, as an active, lethal genetic switch that made their immune system attack their own lungs. We've been told the Neanderthal story wrong. The narrative goes like this. When our ancestors left Africa and met Neanderthals in Ice Age Europe, they interbred and gained useful adaptations. Stronger immune systems, better cold tolerance, survival tools for a hostile new world. An evolutionary gift that helped us conquer the planet. But here's what the latest genetic research reveals. Those same genes that saved our ancestors are now killing us. The Neanderthal variants linked to enhanced viral defense now trigger severe COVID. The metabolic efficiency that prevented starvation now drives diabetes epidemics. The rapid blood clotting that stopped hemorrhages now causes strokes. This is the genetic time bomb. And it's been ticking inside billions of people since the moment we absorbed Neanderthal DNA into our genome. Rewind 47,000 years. Small bands of Homo sapiens are pushing north out of Africa into a world that wants them dead. This isn't the Africa they evolved in. Ice Age Eurasia is a biological minefield. First threat, the pathogens. Every step into this new territory exposes them to viruses, bacteria, and parasites their immune systems have never encountered. Their African-adapted defenses are useless. It's like bringing a knife to a gunfight. And that's just the first problem. Second threat, the metabolism challenge. The food is completely different. No tropical fruits, no familiar plants. Survival means hunting massive, dangerous animals in sub-zero temperatures and storing enough fat to survive months when nothing grows. Their bodies aren't built for this. Third threat, they're genetically bottlenecked. These aren't large, diverse populations. These are tiny founding groups, maybe a few hundred individuals carrying limited genetic variation. When you're facing entirely new environmental pressures with a restricted gene pool, extinction becomes the most likely outcome. But there's a solution already living in these environments. Neanderthals had occupied Ice Age Eurasia for over 200,000 years. Their immune systems had co-evolved with local pathogens. Their metabolism was fine-tuned for extracting maximum energy from fat-rich game animals. Their skin and hair were adapted for brutal cold. When these two human species met, they didn't just compete, they interbred. And the hybrid offspring inherited a genetic toolkit that had been field-tested for 200,000 years. Natural selection went to work immediately. The harmful Neanderthal genes got filtered out over generations. The beneficial ones spread like wildfire through the population. What survived became our secret weapons. The OAS gene cluster produces enzymes that detect viral RNA and destroy it before the virus can replicate. The Neanderthal version is simply better at this job, more stable and more active. When ancient RNA viruses invaded human cells, people with the Neanderthal variant had a preloaded antiviral system already optimized to fight back. This wasn't luck. This was 200,000 years of Neanderthal evolution solving a problem our ancestors would have taken millennia to fix on their own. The TLR gene cluster acts like security guards patrolling your cellular borders, looking for molecular signatures of invading bacteria, fungi, and parasites. The Neanderthal versions are hypervigilant. They detect threats faster and trigger stronger inflammatory responses. For humans entering Eurasia, this was an immediate software update for their immune system, pre-programmed to recognize the specific threats waiting in their new environment. Then there are the keratin genes that build skin and hair. The Neanderthal variants produce thicker, more resilient skin and denser hair. In Ice Age Europe, this wasn't cosmetic. This was survival equipment. Thicker skin provided better insulation against cold and more protection against intense UV radiation at higher latitudes. But the most crucial adaptation involved fat metabolism. Neanderthal genes made our ancestors better at extracting energy from fat-rich meat and storing it efficiently. In a feast-or-famine world where a successful hunt might be followed by weeks of near starvation, this meant the difference between survival and starvation. The pattern is striking. Every major Neanderthal contribution targeted systems that directly interface with the environment: immune defense, skin and hair protection, metabolic adaptation. These weren't random genetic exchanges. This was targeted biological warfare equipment, pre-tested for 200,000 years in the exact conditions our ancestors were trying to survive. And it worked. These archaic genes didn't just help our ancestors survive, they made global human expansion possible. But here's what nobody understood until recently. Evolution doesn't plan for the future. The Neanderthal genes that saved our ancestors were optimized for Ice Age conditions: constant pathogen exposure, scarce calories, extreme cold, and traumatic injuries. Those conditions no longer exist, but the genes remain, executing instructions written for a world 40,000 years gone. Traits that were adaptive then become pathogenic now. The TLR genes that provided hypervigilant pathogen detection now drive autoimmune diseases and allergies. Immune systems calibrated for constant Ice Age threats now attack harmless things like pollen. Your hay fever is your Neanderthal genes treating pollen like a lethal pathogen. People with rheumatoid arthritis, lupus, Graves' disease, celiac disease, and inflammatory bowel diseases carry Neanderthal gene variants at significantly higher rates than people without these conditions. The metabolic trap follows the same pattern. Lipid metabolism variants that extracted maximum energy from game animals now promote obesity and type 2 diabetes. European populations carry 3 times more of these variants. They spread because they prevented starvation. In modern sedentary populations, they increase diabetes risk by 40 to 120%. Carriers develop diabetes earlier and at lower body weights. Factor V and p-selectin variants prevented death from hunting injuries and childbirth hemorrhage. But contemporary humans face 3 times higher risk for blood clots, heart attacks, and strokes. The variants that stopped traumatic bleeding now cause cardiovascular thrombosis in sedentary populations. Neanderthal variants that affect circadian rhythm regulation were adapted to extreme seasonal swings. At Ice Age northern latitudes, winter daylight lasted only 4 to 6 hours, while summer daylight extended nearly around the clock. The ASB1 gene variants helped synchronize behavior with these extreme seasonal variations. Modern life presents fundamentally different challenges. Artificial lighting eliminates seasonal photoperiod variation. Shift work creates irregular sleep-wake schedules. Social demands require rigid daytime productivity regardless of individual chronotype. The Neanderthal variants that helped ancestors adapt to extreme seasons now predispose to insomnia and mood instability. Neanderthal alleles collectively explain between 1 and 2% of genetic depression risk in modern populations. The circadian dysregulation plus chronic immune activation creates a neuroinflammatory environment contributing to major depressive disorder. The evidence gets darker. Some of these genetic time bombs are ticking in unexpected places. A large Neanderthal haplotype spanning the BNC2 gene is carried by nearly 70% of Europeans. Despite this high frequency, suggesting it once provided survival advantages, the variant now significantly increases risk for actinic keratosis, scaly, precancerous skin lesions from sun damage. The mechanism is direct. BNC2 regulates keratinocyte proliferation and skin pigmentation. Neanderthals evolved at high latitudes with weak UV radiation, necessitating lighter skin to facilitate vitamin D synthesis. The introgressed variant likely facilitated this lighter pigmentation and changed how skin cells function. But lighter skin offers less protection against UV radiation. In modern contexts where humans live longer, allowing cumulative damage to accrue, and often inhabit lower latitudes or engage in sun-seeking behaviors, this reduced protection leads to DNA damage in skin cells. This single genetic variant accounts for a significant portion of why some people develop these precancerous lesions while others don't. The genetic variants evolved under low UV selection pressures now increase disease risk under high UV conditions. The circadian mismatch cuts deeper. ASB1 gene variants were adapted for extreme seasonal photoperiods at northern latitudes. The Neanderthal variant appears associated with chronotype flexibility, allowing adjustment to seasonal variations where summer days lasted 20 hours and winter days barely 4. Modern environments present fundamentally different challenges. We experience artificial lighting eliminating seasonal photoperiod variation. We maintain computerized shift work, creating irregular sleep-wake schedules. Social demands require rigid daytime productivity regardless of individual chronotype. The variant that helped ancestral populations synchronize with solstice cycles now contributes to insomnia and delayed sleep phase disorder in environments where circadian disruption from irregular light exposure creates pathology. The circadian disruption connects to something darker. Multiple large-scale genetic studies document significant associations between Neanderthal-derived variants and psychiatric disease. Studies of nearly 28,000 people found that Neanderthal genes contribute more to depression risk than to almost any other condition examined. The mechanism connects to genes controlling your internal clock, immune system, and inflammatory responses. Brain imaging studies demonstrate that circadian rhythm disruption and chronic immune activation both represent significant modern depression risk factors. The hypothesis proposes that Neanderthal variants affecting circadian regulation enhance susceptibility to modern circadian disruption from artificial lighting and irregular schedules, thereby increasing depression vulnerability through circadian-mediated pathways. Another mechanism involves Neanderthal-derived immune variants that keep your immune system in a constant state of alert, pumping out inflammatory signals even when there's no real threat. In modern low-infection environments, these immune-enhancing variants promote chronic brain inflammation that contributes to depression. Supporting this, Neanderthal variants cluster around genes controlling your innate immune system and inflammatory responses, particularly the interleukin signaling pathways that trigger inflammation. Brain imaging reveals the final piece. Higher levels of Neanderthal DNA associate with lower dopamine production in brain regions that control reward and motivation. The pathways optimized for mounting rapid immune responses to acute infectious threats now generate inappropriate inflammation in contexts where infection risk has dramatically declined. And then came 2020, when a novel virus exposed the most dramatic mismatch of all. The pandemic became an evolutionary laboratory. Two Neanderthal haplotypes with opposite effects on the same virus revealed everything we needed to understand about this ancient inheritance. Start with the risk haplotype on chromosome 3, a 50 kilobase segment spanning 6 genes, inherited directly from Neanderthals. 50% of South Asians carry it, 16% of Europeans, near zero in Africans who never interbred with Neanderthals. Carrying this haplotype increases hospitalization risk by 60% and triples the likelihood you'll need mechanical ventilation. The mechanism is a hyper-aggressive immune response creating cytokine storms. When SARS-CoV-2 invades cells carrying these Neanderthal instructions, the ancient genes activate an inflammatory cascade designed for a completely different pathogen from 40,000 years ago. The immune system floods the lungs with inflammatory signals. White blood cells attack infected tissue, but they don't stop at infected cells. They attack healthy lung tissue. The patient drowns in their own immune response. Here's the evolutionary paradox. This haplotype shows signatures of recent positive selection in South Asia, meaning it provided genuine survival advantages against some ancient pathogen. The exact genetic response that protected against ancient respiratory infections now kills when triggered by a novel coronavirus. Context determines whether ancient variants help or harm. But the protective haplotype on chromosome 12 demonstrates the opposite effect. The OAS gene cluster reduces hospitalization risk by 22% per copy. 30% of Europeans and South Asians carry it, 20% of East Asians. The Neanderthal OAS enzymes are simply better at recognizing and destroying viral genetic material. When SARS-CoV-2 enters cells, the ancient enzymes activate, chop up viral RNA before it can replicate, and neutralize the virus before it can trigger the catastrophic immune cascade. We're carrying genetic responses calibrated for viruses that no longer exist. Our immune systems are running software written for biological threats from the Pleistocene. When a novel pathogen appears, those ancient instructions execute regardless of whether they're appropriate for the actual threat we're facing. The effects aren't small. The chromosome 3 haplotype is the single most significant genetic risk factor for severe COVID-19 outcomes identified in the entire human genome. More significant than age-related genes. More significant than any modern human variant. A 50,000 base pair instruction set written by an extinct species determines who lives and who dies when a novel virus emerges. The COVID pandemic exposed these ancient mismatches in real time, but our bodies haven't been passive observers. For 40,000 years, natural selection has been trying to fix this problem. The initial inheritance was massive. Early modern humans carried between 10 and 20% Neanderthal DNA. Today, we retain only 1.5 to 2.1%. Natural selection deleted 80 to 90% of it. The timeline reveals how fast this happened. Most purging occurred within 100 generations of initial contact. That's roughly 2,500 years. By 45,000 years ago, only a few centuries after the first interbreeding events, the major introgression deserts were already evident. Ancient DNA from individuals living 45,000 years ago shows substantially higher Neanderthal ancestry than modern humans, proving the purging process was immediate and ruthless. The chief mechanism was population size. Neanderthals lived in small, isolated bands for hundreds of thousands of years. In small populations, natural selection is weak. Slightly bad genes accumulate. The Neanderthal genome was full of them. Modern humans had much larger, interconnected populations. When Neanderthal DNA entered the human gene pool, those slightly harmful genes faced intense selection. What was tolerable in small Neanderthal bands became intolerable in large human populations. But the purging wasn't uniform. Some regions became introgression deserts, vast stretches where archaic ancestry is completely absent. These deserts reveal the functional boundaries where Neanderthal and modern human biology proved fundamentally incompatible. The X chromosome shows the most dramatic depletion. Only 0.3% Neanderthal ancestry compared to 1.4% on other chromosomes. A 76% reduction from expected levels. Male hybrids carrying Neanderthal X chromosome genes likely suffered fertility problems, triggering swift elimination to restore reproductive viability. A massive region on chromosome 7 containing the FOXP2 gene, the language gene, is entirely devoid of Neanderthal DNA. While the protein itself is identical between species, the switches controlling when and how much the gene turns on differ significantly. The Neanderthal version disrupted how modern human brains develop language circuits, likely impairing speech and language ability in hybrids. Testis-expressed genes show extreme depletion of Neanderthal ancestry across the entire genome. This parallels the X chromosome pattern, suggesting reproductive incompatibility was a major selective constraint. Neanderthal variants likely disrupted sperm production and reduced male fertility in hybrids. The survivors tell their own story. Variants that persist either provided strong benefits or caused problems only in modern contexts. The OAS genes protecting against viruses remained because viral defense provided ongoing advantages. The hypercoagulation variants remained because childbirth complications continued killing women well into historical times. The metabolic variants remained because food scarcity persisted until industrialization. Selection continues today, but environmental change outpaces evolutionary response. We're running a modern biological system that still relies on patches of ancient, deprecated code. While these patches saved the system during the volatile Ice Age release, they've now become technical debt, a source of bugs, glitches, and system instability in the modern environment. The key realization is simple. There are no inherently good or bad genes, only genes matched or mismatched to their environment. The genes that allowed humans to conquer Eurasia now contribute to our most prevalent chronic diseases. Understanding this archaic legacy is no longer just anthropology, it's clinical necessity. Recognizing that a patient's risk for diabetes, clotting disorders, or severe COVID may stem from a 60,000-year-old encounter allows for targeted risk assessment. Personalized medicine must account for the fact that we're all carrying genetic instructions written for a world that no longer exists. The question isn't whether we've evolved beyond what Neanderthals were. The question is whether we can adapt faster than our ancient inheritance can harm us. If this journey into our genetic past left you questioning what other evolutionary compromises are written into your DNA, subscribe to explore more of the uncomfortable truths buried in our biology.